Zoom.
Zoom.
Yeah. Okay. I mean,
Okay, sir, can we start?
yeah.
Yes.
Yeah, we can start.
So I have
Prasanjit,
already started the
recording?
YouTube,
You start recording
and
here?
2 minutes left. It's not
Okay.
9 yet.
Okay. It's
So
2
I
minutes.
heard from Arpon that Dr. Bush is now in California.
No, I'm actually in Iowa.
Okay.
So what's time there?
Yeah.
Is it
It's 10:30 p.m.
10:30? So is it comfortable for
10:30.
you?
Yeah. Yeah.
Oh,
I don't have a
fine.
problem. I stay up pretty late.
Okay, that's fine. Okay, so just 1 minute left. I'll start the recording.
Okay.
So I'm starting the recording. Okay, Shilajit?
Yes.
Okay, sir.
1 minute.
Wait,
1
now?
minute. 1 minute.
1 second.
Just look.
Yeah, you-
Yeah, now start.
You can.
Okay. A very good morning to all distinguished participants, my respected teachers, my fellow research scholars, and my junior students. And a very good evening to our speaker. I know it's night, but still, this is Anushree Hai, research scholar at the Department of Microbiology, Vijayagarh Jyotish Roy College. I heartfully welcome you all to the session 3 of International Webinar Lecture Series in series of origin, prevention, and possible cure for COVID-19. That is organized by Department of Microbiology in collaboration with Internal Quality Assurance Cell, Vijayagarh Jyotish Roy College. Eminent speakers in the last 2 sessions had enriched us with molecular distinctiveness and therapeutic implications of SARS-CoV-2, the causative agent of the COVID-19 pandemic, which the world is currently experiencing. We have reached the final session of the lecture series, and we are eagerly waiting to hear for other implications of COVID-19 pandemic. This lecture is being telecasted live in YouTube, and we would humbly like to request our participants to drop down questions relevant to this lecture in the chat box. Feedback link would be made available to all of you and need not to be worried for it. First, I would request Dr. Prasenjit Das, the head of the Department of Microbiology, Vijayagarh Jyotish Roy College, to deliver his welcome address. Sir, please.
Thanks, Anushree. Good morning, everybody. Is it getting echoing?
Yeah, it's,
Yes, sir.
Sir, Shantan sir, please could you mute your audio? Then it will be it's echoing from
Okay.
the Shantan sir's audio.
Okay.
Sir's audio may be already muted. I mean,
No. Sir is in 2 windows. Yeah. Now it's okay,
Oh, yeah,
sir.
right.
It's okay.
That's okay. So small technical glitches associated with these webinars. So this is Dr. Prasenjit Das, Head Department of Microbiology and Joint Internal Quality Assurance Cell Coordinator of our college. Due to some technical issues, our respected principal madam is unable to join this program. So on behalf of the department and Vijayagarh College as well, I would like to convey my heartiest welcome to the speaker for this session, that is Dr. Shantan Bose from Autonomous Therapeutics, J Labs, New York, USA. As it was already mentioned in the flyer, his topic of today's talk is the SARS coronavirus, COVID-19 and our future. So I guess this talk will mainly focus on the therapeutic aspects of SARS-CoV-2, which is in parity with the main theme of this webinar, that is in search of origin, prevention and possible cure for COVID-19. Dr. Bosch is an expert in the field of therapeutics, and I guess that this is going to be an exciting session for all of us to listen to his talk and interact with the speaker as far as possible. You can put your questions in YouTube message box. I also welcome the participants coming from different educational institutes across the state of West Bengal and beyond, who are here this morning to listen via YouTube live streaming. Uh, please put your queries regarding speaker's talk in the message box. We will convey the messages to the speaker during the question answer section of this session. Please do interact so that the session will be more exciting. So now it's over to Anusri to proceed further with the program. Thanks.
Okay. Thank you, sir, for your warm welcome address. Next, I would subsequently request Ms. Shahana Ghosh, the ACCT of our department, that is Department of Microbiology, Vijayagarh Jyotish Roy College, to formally introduce our respected speaker for this session. Shahana?
Yes, am I audible?
Yes, you
Yes,
are.
Shahana.
Thank you, Anushriti. A very good morning to all the participants and respected teachers present here, and a very good evening to our respected speaker. It is my great pleasure to introduce Dr. Shayantan Bose, who is a senior scientist virology at Autonomous Therapeutics Inc., New York, USA. Dr. Bose has completed his Bachelor of Science in Physiology in 2004 from Presidency College, Kolkata, under the University of Calcutta, and ranked third across the university. Then he completed MSc in Life Science in 2006 from the School of Life Sciences, Jawaharlal Nehru University, New Delhi. There he ranked second in life sciences across the university. After that, Dr. Bose completed his PhD in biological sciences in the year 2012 from the Interdepartmental Biological Sciences Program, Northwestern University, USA. He was postdoctoral research fellow at the Department of Molecular Biosciences, Northwestern University, Evanston, USA from December 2012 to December 2013, and his research was on investigating molecular mechanisms of paramyxovirus entry. Then Dr. Bosch was a research fellow from December 2013 to February 2020 at Microbiology and Immunobiology Department, Harvard Medical School, Boston, USA, and his research work was on investigating viral host entry factors. He was he has several publications in highly ranked international peer-reviewed journals and received many awards and professional grants also. He was nominated for the prestigious President of India Gold Medal from the School of Life Sciences, Jawaharlal Nehru University, New Delhi in the year 2006. He attended many conference presentations and workshops and teaching experience as well. Dr. Bose acted as an ad hoc reviewer for major scientific peer-reviewed journals. Now I would like to welcome Dr. Shantan Bose for delivering his lecture in today's webinar on the topic the SARS coronavirus COVID-19 and our future. Thank you, sir. Now over to you, sir.
Sir, could you please unmute your?
I think sir is here.
Yes.
Yes, sir is there. Sir.
Okay.
Yes, sir is here.
It's visible.
Can you hear me now?
Yeah.
Yes, sir.
Yes.
Yes, sir.
Absolutely fine.
Okay.
Thank you, sir.
Oh,
But,
looks like the camera
some
is working.
echoing is there.
Yeah.
Yes, sir.
Yeah,
We
yes,
can
sir.
see you too.
Acha,
Okay.
all organizers, please mute your microphones because they are. Yeah. Thank you, sir.
Okay. I think the camera is creating some sort of issue, so I'm going to turn it off because it's creating some kind of background. Okay.
Yeah, it's okay.
Okay, so I'm going to start my presentation by showing you this series of headlines. So what I've been putting this together because this is kind of what we've been seeing in the news every day. There's a ton of headlines, lots of big news. Things are shutting down, and it's scary. I mean, everybody understands that this is really scary, and like most of us are very disturbed by these kind of headlines. So what I would say is that I would start off with this quote from The Art of War by Sun Tzu, who was a military general in ancient China. It says that if you know your enemy and you know yourself, you need not fear the result of 100 battles. So, my point is that we really need to know this virus in order to not be scared of it. So, this is not something new, this is not something alien. Uh, so, the best way to deal with the stress of the current scenario is to think like a virologist. And this is something from a personal point of view as well, that in this entire scenario, I have been really calm because I understand exactly what the virus is doing. And once you get into the detail of the biology, all the virologists, they will be much more comfortable dealing with this virus. So I want to share that with you today and focus on the biology of the virus, look at some of the headlines, look at what is going on, and seeing what exactly is happening around the world in all of these headlines. So in my talk, I'm going to kind of distribute it in these 5 different sections. The primary part will be the biology of the virus, because I mentioned that if you understand the biology of the virus really well, you will understand the rest of the other aspects as well. So starting off with this, a couple of questions here. What is the novel coronavirus? How does it infect us, and where did it come from? Again, looking at these headlines which have been coming up in newspapers all over the world, it's a killer virus, it's a novel virus, it's an alien virus. Is it really that? It is a novel virus for sure, but it is a member of the coronavirus family. So, as a department of microbiology, you'd be pretty familiar with this family, and you'd be pretty familiar through your studies as well as through news in over the last decade or so. And you would have heard about the MERS coronavirus, the Middle Eastern Respiratory Syndrome virus, the SARS coronavirus, the original SARS coronavirus. And then you might not have heard that much about these other viruses that I have on the screen. So OC43, NL63, and 229E coronavirus, these are all part of the same family. But in fact, these viruses are way more familiar with us than we are familiar with them, because these are normal common cold viruses that happen every year. So pretty much all of us have at some point of time in our lives have been infected by one of these viruses. And they are all coronaviruses. So they are part of the same family, yet we never know about them because they are not that dangerous, and nobody dies of these viruses. All of these viruses shown here, except MHV, is a mouse hepatitis virus. All of the other ones are human viruses. So SARS-2 coronavirus, that is what's the current pandemic is going on. It's a newly discovered member of a very well-known, very well-studied virus family, and we have been studying this virus family since the 1960s. And as I mentioned, we have been infected by various coronaviruses since the beginning of humanity, but only epidemics starting in 2003 for the original SARS coronavirus, the MERS coronavirus in 2012, and now this coronavirus has brought this relatively unknown family into prominence in recent year. In fact, before 2000, hardly anybody studied coronaviruses because they were not considered important. So coronaviruses are single-stranded, positive-sense RNA genome, as you would know probably. And this is just to isolate, this is just to show you that this novel coronavirus or coronaviruses themselves are part of the entire virus family, RNA virus family, and they are not something unusual, alien, or unknown. They are part of this group called, order called Nidovirales. And I've just highlighted some of the known other orders that we are familiar with, like the flu viruses come from orthomyxoviruses. Mononegavirals are a big group which has measles, mumps, Ebola, and other families, which these are representatives of. We know of the chikungunya virus. Zika and dengue virus come from the flavivirus supergroup. So overall, this basically shows you that they are part of a larger family, and they just happen to be a certain group of viruses out of which only one virus currently is dominating everything, or the news, the world, and all of our lives. So, how novel is this virus? So, as I mentioned, the SARS virus caused human deaths in 2003. And this particular SARS-2 coronavirus has just not infected humans before. But when you look at these genomic alignments of the viruses, you can find uh, SARS coronavirus sits pretty well close to between a group of bat coronaviruses. So they are very closely related to bat coronaviruses, pretty closely related to the SARS coronavirus, and a little further away from the MERS coronavirus. So going a little bit further into this phylogenetic tree, what you can see is that bat coronaviruses, of course, they are like some of the closest members, and some of them only have a few sequence changes compared to the SARS coronavirus, which suggests that these must have come from one of these viruses. So how did it come from these viruses? So here I'm introducing a term which you might be familiar with called zoonotic viruses. Zoonotic viruses are viruses that jump species to infect humans. So they are originally animal viruses. They are present in nature. They have been going around forever, but we just don't know about them. And we only come to know about them when a human gets infected. And how does that happen? The fact is that this particular type of jumping species has happened quite a lot over time. So in recorded history, we know a lot of these viruses which actually came originally from animals. For instance, Marburg viruses, the Ebola virus, Nipah virus, which was actually prevalent in the eastern part of India and Bangladesh. SARS coronavirus, H5N1 bird flu, and all of these other viruses, which, as I show here, they came from these different animals. So viruses can start infecting human host and jumping from these different animals. And bats, as you can see in a lot of these cases, are that certain animal which harbors these viruses. And bats have a very unique biology that prevent them from getting sick. So that's why they can carry these viruses, but they don't get sick by them, and somehow they can transmit that to humans. Now, bats and humans have very rarely come into contact unless you actually go into caves and get in contact with bats. But oftentimes, what happens is other animals that live in caves with bats, they get infected by these different viruses, and these animals have a more closer connection to humans, as like they might come into human habitats, and they have much stronger interactions. They could bite humans, and that's how interspecies route of transmittance can happen. But then, how does this jump actually occur? For this, we go a little bit more detail into the molecular biology of the virus. And as I mentioned, these viruses are enveloped positive stranded RNA viruses. They are, as on the left, you can see a EM image of the virus. And basically on the right is that reconstruction from that image of the virus with its various proteins highlighted. The genetic material of the virus, which is RNA and its positive stranded RNA, is coated by a protein which protects it from the cellular RNA recognition proteins, which can damage it and destroy it. And then that's coated, it's covered by an outer layer of matrix protein, which is covered by another layer of membrane, which is derived from the cell itself. Now, this membrane has a bunch of different envelope or glycoproteins on its surface. Today, I'm just going to focus on one glycoprotein because that is the most important one, and you'd probably realize why. Because that is the spike protein, and it's a huge glycoprotein. It's which gives the virus its name. So it kind of sticks out from the membrane and provides and looks like a crown. So crown meaning corona. So that's where the name comes from. Looking a bit further into the molecular structure of the spike protein, this is what it looks like under electron microscopy. And further, you can see that this is how these viruses get into cells. The spike protein surrounds the entire virus and is the first protein that comes in contact with the human host cell. So basically, that is the protein which recognizes whether or not a virus can enter a cell. And this it does by recognizing a protein on the surface of cells. And this protein, as you would probably know, is known as a receptor. So each virus must typically engage with a very specific protein or carbohydrate molecule on the cell, which is a receptor, and each virus has its own receptor. Now, this is very important because a virus can only enter cells which express its specific receptor. This will also decide then that which host and which organ it can infect. And a receptor, interesting part is that receptor is not basically built for a virus to get into cells, because what cell would really want that? So cells don't design these proteins to invite viruses in. Viruses, on the other hand, during evolution, usurp or take advantage of these cellular molecules which kind of stick out from the surface of cells and use these as receptors to enter cells. The specific receptor for the SARS-2 coronavirus is the angiotensin converting enzyme 2, or ACE2. So I'll talk a bit more about that in detail and why this is important, and going a bit further into the biology of that. But for now, this is just a table. It's not important like what each one is, but this is just to highlight that all of these different viruses have their own receptors, and they could vary from proteins like ACE2 or DPP4, which is the receptor for MERS, to something like a carbohydrate, like sialic acid, which is a receptor for influenza virus. So now that we know that there are certain proteins that the virus uses to enter cells, and these proteins are the ones which undergo mutation, and pretty much all of the proteins of a virus undergo mutation, but the mutations in this protein, the external glycoproteins through which the virus enters, is the important one in terms of what it can infect. And so when those proteins mutate through natural mutation rates, which are kind of like 10 to the 6 to 10 to the 4 in nature, uh, then a lot of things can change, and we'll talk a bit more about that now. So usually the normal mutation rate for RNA viruses, because RNA is a molecule that can be changed easily, unlike DNA, because DNA has two strands, and so it has to copy the exact same strand. And there are repair mechanisms which can change one strand with respect to other and correct any mistakes that can be made. But for RNA viruses, which just have a single strand of RNA, it's really easy to make those changes, and it's really easy to mutate the virus. So as a result, viruses kind of thrive on this mutation. They keep on changing. As some of them die out, most of them die out, in fact, but eventually some of them change into something else, so they can infect different cells, they can infect different animals and different hosts. So what has happened, and what has happened for this virus has happened for many, many other viruses over history and in time, that certain mutations in the spike protein have come up in this bat-like precursor virus of this SARS-2 coronavirus. And these, initially, this bat SARS-2 virus cannot infect human cells because it cannot bind to the human ACE2 receptor. But it might be able to bind to the bat ACE2 receptor. But there are certain changes in this human SARS-2 virus, which is the current circulating strain, which now allows it to bind to the human ACE2 receptor. And so this mutation happened just by chance. It happened in maybe some intermediate animal or in the bat itself. but somehow it got transferred to humans, and now that's the reason why we have this virus infecting us. So the good thing is that the vast majority of these mutations that keep on happening are really bad for the virus and makes it less effective. So because we design vaccines, and because I've had a lot of experience in working with the genomes of viruses, it's really difficult to get a virus, to make a mutation in a virus and allow it to produce a good replicating virus, because any mutation you try to make in a virus, it kind of dies out. So most of the mutations, I would say 99.99% of the mutations make the virus really sick, and it kind of dies out. But certain mutations like this one, which I just described, might just change its host and allow it to infect different cells. So uh, we actually have, this is a very recent report that came out I think last week or week before. We've actually seen virus evolution happen right before our eyes, because this coronavirus, which initially started out and was discovered at the end of last year, it had a mutation, or it had a sequence in its residue aspartate at position 614. That was the original virus. Then something happened in February where, just by random changes, suddenly this changed to a glycine. This particular residue changed to a glycine. And now that virus, the new virus, the mutant virus, is now so much more infectious that it's taken over the entire population. So right now, the original virus that we started off with in last December is not there anymore, or it's in a very small number of individuals. Most of the viruses that are circulating for SARS coronavirus 2 is now this new mutant. But I mean, the good thing is that this mutant is way more infectious, so it can infect a lot more people, and it can spread more rapidly in the population, but it's not necessarily more pathogenic. So that was a saving grace. And even though the virus is spreading way more now, uh, it it is not, affecting, or it is not causing a lot more deaths than the original virus. So just to sum up with the spike protein, the reason why it's so important, and it'll come up again in my presentation, is that firstly, it's a major protein that's used for infecting cells by the virus. It decides which cells of the body are infected. It decides the host which is infected. It's the major protein that remains outside the virus, so it comes in contact with the cell first, and rapid mutations in this protein can cause the virus to change its host or receptor. And also, which will become important in the next section, it's a major target of natural immune responses of the body, and one of the major targets for developing vaccines and therapeutics. So I'm going to talk a bit more going into the medicine part of it, or which is basically the pathogenesis part of the virus. How does it cause disease? What is COVID-19? So, how does SARS coronavirus 2 cause disease? And why are older people and people with comorbidities more susceptible? Again, we've been seeing all these headlines, and like all of us know about these comorbidities, what these are: cardiovascular disease, diabetes, kidney disease, which can really worsen the disease of COVID-19. So, how does that happen? So here, I mean, you would have noticed that I've been calling the SARS-2 coronavirus, I've been referring to the virus as the SARS-2 coronavirus. And then now I'm switching to its disease, which is COVID-19. It is basically a short form of coronavirus disease 2019. So right now I'm going to talk more about into the disease aspect. So let's start with the receptor. So remember that the receptor of SARS coronavirus 2 is ACE2. It's angiotensin converting enzyme 2. This receptor is present in a lot of cells in organs like lungs, the GI tract, heart, and the circulatory system. They just happen to have a large number of ACE2 molecules present on their surface. So naturally, that provides these cells to be the major target of the virus. So what does the angiotensin converting enzyme do in the body? Remember that the receptors are normally, they have a normal function in the body. for usually. So angiotensin is a hormone in the body which is required for vasoconstriction, and it increases blood pressure. This is basically it's kind of like a emergency hormone. So in terms of like when there's wound or when a there's some damage in tissues, this hormone is secreted, and it brings in a lot of white blood cells, which can then help repair those damaged tissues. So too much angiotensin will cause overactive white blood cells entering tissues, but then these white blood cells, our own cells, will cause damage of the tissues because they are damaging the normal tissues, and they will release a lot of inflammatory small molecules known as cytokines. So this, I mean, uncontrolled levels of angiotensin is bad for the body. It is only required when there's damage. So ACE2 is the enzyme that breaks down this excess angiotensin to prevent the above effects. So in effect, it kind of protects the body from the overactive angiotensin function. But then the virus just happens to bind ACE2, and this prevents it from carrying out its normal function. So most of the ACE2 is taken up by the virus, while the angiotensin levels keep on increasing. And when angiotensin levels increase, there's increase of blood pressure, inflammation, and tissue damage. So that is what we are actually seeing in patients for COVID-19. You know that alveoli, they are structures in the lungs which help us exchange oxygen and carbon dioxide. Uh, the cells that line the alveoli have very high ACE2 expression. So what happens is that when virus, it enters normally through the lungs, and it binds to these cells in the alveoli and trigger the immune response, as a result of which a bunch of different immune cells come into the lungs and block out the alveoli. As a result of this, there's a block in the oxygen exchange, and that's why the patients who are actually very severely affected by COVID-19, they are oftentimes dying as if they are drowning. So a lot of like postmortem reports have shown that the lungs look like they have been from a patient who has drowned underwater. So that's the respiratory effects that we've been seeing. There's also inflammation in the lungs, as I mentioned, and the immune cells that are responsible for this inflammation. So these cells will release a lot of cytokines, and cytokines are small molecules which will trigger a lot of different effects, such as atherosclerosis leading to strokes. So a lot of these effects, like it's a general response of the body that will cause vasoconstriction, atherosclerosis, and angiotensin also has a major role in forming blood clots. So it can, if it causes clots in some major arteries, that could be a major issue. So together with all of this, angiotensin kind of damages the entire circulatory system, as well as the lungs and the parts of the alveoli. So this is what the doctors have been seeing in COVID-19 patients, and this is quite unusual because usually the virus doesn't have a systemic effect like this. So it's quite unusual that the SARS-2 coronavirus is having this effect, even though the SARS-1 coronavirus hasn't been studied to that detail. Maybe further studies will be done after this. So the question is then, how does our body respond to a pathogen like SARS-2 coronavirus? And then again, I'm bringing out this headline that we might have been seeing for the last few days, that immunity to this coronavirus may only last for a few months. There's this recent study that came out. And as a result of this, a lot of people are asking, so is the vaccine going to be effective then? Because if we don't have antibodies, how are you going to combat the vaccine won't have much effect anyways for a few months. So the fact is that our bodies are constantly exposed to foreign matter every minute of the day, and they generate antibodies constantly. And generally, like everything that you come in contact with and you haven't been in contact with before is a foreign matter. So every time antibodies are generated, they remain in circulation for a few months to a few years. And then if the pathogen or whatever the foreign body is not encountered again, the antibodies actually disappear. And this is completely normal. So you'd be familiar with this antibody response, that initially when you have the first infection, you have a naive B cell. B cells are the cells that produce antibodies. These B cells then become activated and start producing antibodies. Now, this stays on for some time, but then later when the infection goes away and the body does not encounter that infection anymore, the cells that produce the antibodies, that those specific antibodies to that particular specific virus infection, will go down. But that does not necessarily mean that the overall antibody level of the body has gone down. It's just that for certain specific antibody for that particular virus or bacteria or whatever foreign particle has just gone down because you don't need it anymore. But even though it goes down, there is a certain group of memory B cells, which are known as memory B cells, which retain that information that there was this particular antigen or that we had been in contact with. So these memory B cells are few, but they remain in the body for a very long time. And if there is a repeat infection of that exact same thing, then these memory B cells are triggered again, and they start churning out these tons of similar cells to themselves. And as a result, that same antibody which was initially created is made in much more larger amounts again. So this is the secondary antibody response. So we must always remember that vaccines rely on memory B cells and memory T cells as well, which I'm not getting into in this presentation. But these two are the most important factors for a memory B and for a vaccine to work. So when the newspaper reports are telling us that we don't see antibodies for the 3 months or 6 months or whatever, it does not necessarily mean that the vaccine is not going to be effective. As long as the memory B cell response is being generated or memory T cell response is being generated, we should get a vaccine which is effective. And in fact, this is a very interesting study that has been recently published, that there were many memory T cells that were found in people who were unexposed to SARS-2 coronavirus. So people in countries which had never seen this SARS coronavirus, people were seeing memory T cells which have this, they are very specific to the SARS coronavirus. So what has happened here is that, remember those other viruses that I talked about, which are common cold viruses, but they're still coronaviruses, OC43, NL63, 229E. Well, those coronaviruses are quite similar to this current SARS coronavirus. So when people get infected with these viruses, the body just takes up pieces of these viruses, chops them up, and the T cells keeps a record of what has happened and which virus has infected us. And some of those fragments of these different other common cold viruses match that of this new SARS coronavirus. So as a result, any antibody that is created against those common cold viruses are now effective against this new pandemic virus. So similar viruses oftentimes have cross-reactivities, and we have seen it for a lot of other viruses as well. So in virology, it's a big subject of study. So again, this is highlighting the fact that SARS-2 coronavirus is not a unique virus. It just happens to be related to a lot of other viruses, and we might even have immunity to some of it. And so this is another report which just came out maybe 2-3 days ago. It claims that the vaccine that the Oxford group has been developing, it provides a double protection against novel coronavirus. So basically, that's what it means. It means that they have found T cells, which are which memory T cells, which are getting activated. So or which are getting stored in the body. So which means that on the long term, these T cells can come back and fight if the person gets reinfected with SARS coronavirus 2. So so that's how like I'm trying to interpret the headlines and like what they actually mean, and hopefully that'll give us some clarity of like what's going on in the world and not get overwhelmed by too much of the news. So now moving on to the other section, vaccines and therapeutic development. This is actually epidemiology. Yeah, so we are moving into the epidemiology section, asking how is this virus spreading from person to person. So now we must, by now we are pretty familiar with what are the different ways this virus can spread. One is through surfaces, uh, touching surfaces for touched by an infected person, followed by touching the eyes, nose, or mouth. These are the ways that the virus can get in because they are mucous membranes, and this virus can get in through mucous membranes. And the virus can last on various surfaces for hours and days. We've hours to days. So we've seen a bunch of studies regarding that. And then we've seen a bunch of studies regarding the droplet spread. So an uncovered sneeze that can propel droplets at about 200 miles an hour can go up to 23 to 27 feet based on a study at MIT. And the other way that it can is being acknowledged that it can spread is through airborne transmission. So what happens is that when a person sneezes, the virus particles are coated by mucus, and these are pretty heavy. So because they're coated by mucus, and that can't travel too far beyond up to maybe 6 feet. That's why the regulation of keeping apart for 6 feet. Then, as these particles travel through the air, the mucus starts to evaporate. And then these particles start getting smaller and smaller. So these smaller particles can now travel up to 27 feet. But then when the mucus evaporates completely, what is left is known as infectious droplet nuclei. These can remain in the air for a very long time. But on the other hand, because there is no protective mucus coating, a lot of these particles are actually damaged. So the chance of getting infected by a sufficient number of these particles drops considerably when we go into the airborne transmission phase. So even though airborne transmission sounds very scary, and we've been hearing a lot about that, a lot of those particles, you have to get a very high concentration of very infectious particles in order to get infected by airborne transmission. So with the chance is pretty low for that. And then we've also talked about R0 value or R0 values, which represent the number of new infections that stem from a single case. The important point here is that given all persons can be equally infected, so if all persons cannot be equally infected, which means that these people have some kind of immunity, the R0 value of the virus can be brought down. And this chart just gives you an idea of like how other viruses have an R0 R0 factor. Measles, for instance, is one of the most contagious viruses, human viruses known to man. It can infect up to 18. One person can infect up to 18 individuals. So compared to that, this virus, the coronavirus has the R0 factor is about 2 to 2.5. So we can see that in comparison with other viruses, it's still not that infectious, and maybe the newer mutant version is a bit more infectious than this. What this value shows here on the slide, but it hasn't been calculated as of yet. So then now we move into the vaccines and therapeutic development. So the question that everybody asks is, how long is a vaccine really going to take to develop? So here I have a picture from New York Times that came out, which I illustrated this pretty well. This is the traditional scenario for a vaccine development. You first do academic research. There's preclinical research, includes some animal testing. There are 3 phases of the trials, which are all human testing, in increasing number of individuals that have been tested in each phase. But every phase has to be completed before the next phase starts, because you don't know how the humans are going to react in the different phases. Then you start building factories, which take a long time. Then you start your manufacturing, then there's the approval process, and then distribution. So if we went by this traditional scenario, starting from 2020, the vaccine would take 16 years. And this would mean that it would come out in 2036. But of course, that is not an acceptable situation in this case, given that it's a pandemic and what the situation the world is in. So what other vaccines have taken? Like how long known vaccines have taken? So things like dengue or Zika vaccines have taken really long since the virus has been discovered. Um, Ebola vaccine has also taken about 20 years. But in recent times, the MERS coronavirus vaccine has been pretty short. So it's taken about 22 months. But really, what we have to do for the coronavirus here is much more shorter than that. And how do we do that? How do we accelerate this process? So the need right now is basically compressing the whole vaccine development process into few months. That 16-year process goes into few months. As a result of this, we have to skip a lot of research. There's some of the basic research that's normally carried out. Uh, Clinical trials, as soon as some safety trials have been done with animals, human trials will have to be started. Number of phases have to be combined. And in fact, factories are going to be built even before the phase 1, phase 2, phase 3 trials have been completed. So people don't even know what vaccine are going to be is going to come to the market, but they are building factories. So they are building factories based on 4 or 5 different vaccine types. So whichever one works, they're going to start making it in those factories. Manufacturing is going to start quickly. Approval will take much less time, and then distribution. So this is what we have to do. And basically, this is just showing how that entire 16-year process has been combined into 1.5 years at this point. So already, this is kind of an all-in approach. And there are issues with this, of course, that it must be evident to you when I was showing the data before, that a lot of vaccines have been developed without proper in-depth research. There's a massive chance of failure. There could be a safety issue, and then the whole endeavor will require a massive international cooperation, and it'll come at a huge cost. But the fact is that at this point, very little choice is left. But the good thing is, more than 155 different types of vaccines are in development simultaneously. So all different types of vaccine platforms, and some of them are very well tested, well-known vaccine platforms, which uh, based on which other vaccines have been developed. So we know that those are safe. We know that they can be given to humans. It's just building a vaccine based on that platform. So as you can see in this diagram, 14 vaccines are in phase 1, 11 are in phase 2, 4 are in phase 3, and then one vaccine has been approved for limited use for military in China. So in addition to these vaccines, about 254 therapies have been explored. So based on this, I mean, this has never been done before. So based on this, we can definitely be hopeful that there would be vaccines. One or in fact more of these vaccines or more of these therapies are going to work, and we would very soon have something that will prevent COVID-19 disease. So coming to that, what is the future going to hold for us? What will it? How will we deal with this epidemic, and how will our lifestyles change after this outbreak? This is the section that I'm going to go into now. So many of these short-term emergency measures that you have seen recent times may become a fixture of life, that it might be very normal nowadays. Like if you're going to a large gathering, say, let's say if you're going to watch a cricket match, or once you enter the stadium, there might be infrared remote body temperature detection, which you might not even realize. But like if you have a fever, or if you have some slightly of temperature slightly above what is recommended, there you might be pulled out and you might be like tested. There could be touchscreen monitoring. So whenever you are checking in, you're touching the screen, it'll record your temperature through your finger. Then you might certain persons who are at risk based on their age or based on their comorbidities they could have an at-risk certificate, and certain persons who are tested positive, they might already have antibodies circulating, so they could have tested positive certificates. Also, there could be health monitoring bracelets, which can constantly send data for high-risk individuals to make sure that they are not running a temperature, not falling sick. And then, of course, the social distancing measures that we have all seen. There could be one-way walkways, 6 feet distances, plexiglass partitions, and less density of seating in public places. So now, what I'm coming to is is a really important aspect, which is going to be the primary importance of preventing a pandemic in the future. So there's a lot of scientists have over the years, and this is a very big field in virology, and it's grown bigger year over year. Uh, it's called virus surveillance. It basically involves going around into different habitats of animals like caves or forests or fields, and then just sampling for animal droppings from caves or trees, and then sequencing all of these animal droppings, because now that we have the capability to sequence a large amount of data, we can sequence these small genomes of viruses really rapidly. We can sequence hundreds and thousands of these viruses from single samples. Uh, so, so these virus hunters, the scientists that they like call themselves, they would look for all of these novel viruses of known families. So remember that coronavirus family was a known family, but the virus was an unknown family. So it's basically looking for such viruses by random sampling in animals. Now, I picked out this particular study for a very certain reason, because these kind of studies have come out for years and years, and really they haven't received any attention. But see, if like we had paid attention to this study in 2012 when it was published, we would have found that in Hubei province in Wuhan, there were bat coronaviruses which were very similar to that of SARS coronavirus. So these had already been isolated from bats in that area in 2012. But since there was no government funding or nobody really cared, so this wasn't followed up again. And it does not necessarily, it's not necessarily for China, but it is also true for literally everywhere around the world. In this slide, I'm showing you multiple studies that have been carried out all over the world. The first one by an Australian group in different continents in 2011. They found a huge number of mammalian paramyxoviruses. Then in Kazakhstan, into 2019, they found bat coronaviruses. In Vietnam, they found paramyxoviruses and coronaviruses. Just for reference, paramyxoviruses is the family from which Nipah virus comes. So all of these would potentially have deadly viruses which can jump from these animals into humans. And then the last study is in US, Canada in 2017, which found another large group of bat coronaviruses. So other than these studies, there's another way that scientists try to predict what could be the next big pandemic or epidemic virus, is to try to mathematically model how a virus sequence can change. So once we get all the data from these other studies that I just described, then people try to do mathematical modeling and see what could be the potential sequence changes of these viral genomes, and what could be the next virus that will make it really, what made the next mutation that could make it really pathogenic. So, and based on that, I wanted to show you a success story which just recently came out, and you might have seen this news again taking again from another headline. This news came out in June, at the end of June, which said that a new strain of flu was found in China, which has a potential to become pandemic. So this flu strain doesn't infect humans yet, but the scientists have found that it has a chance to mutate into a strain which can now affect humans. So the CDC, Centers for Disease Control in the US, has taken rapid action against this. and is now studying this strain. So this is a success story because it may sound scary that there are lots of viruses that can become pandemic, but if we find them and if we can predict them, then we can take action against them even before they come into the human population. So basically, to sum up, we need to have trust in science and scientists. We need to have more representation of scientists in political positions. There needs to be a global sharing of data, which is really important, and a global effort for redistribution of resources and reallocation of resources. So just to summarize this, proper surveillance, predictive technologies, governmental cooperation, and our knowledge, which is the most important part, of such potential pathogens can prevent the next COVID-19 pandemic from happening. And so I just have some resources that are pretty reliable resources for getting information about coronaviruses. And I would like to thank you all, and a special thanks to Professor Prashanjit Das, the head of Department of Microbiology. I'd like to thank the entire Department of Microbiology and Vijayanagar Jyotish College, Kolkata, for inviting me to this talk. And it has been a really big privilege for me, and it's been wonderful to speak to you. And I thank you for your attention. Have any questions?
Thank you, sir.
Ankita, before the question and answer session, I have an announcement. Volley of questions are waiting for Dr. Bose, I guess. Before that, I would like to announce that our principal madam is in YouTube. She is watching the whole program, and she has written some messages that I hope that this session will be immensely helpful and very informative. Thank you all. Stay safe as much as possible. So, thanks, madam, for your encouragement. You are always with us for arranging such kind of seminars, or rather webinars. So, very good morning to you too, madam. Ankita, you may start now.
Okay, am I audible? Hello?
Yes,
Yeah,
yes,
yeah,
you
you
are.
are.
Am I audible?
Yeah.
So, thank you so much, sir, for such an informative presentation. So, now I'm proceeding with the interaction session. So, the first question is from Shubham Bodi. He asked, how can asymptomatic patients spread the infection?
Okay. So that's still something that's been investigated so far, but there's one correlation that's been seen with a lot of these patients who are asymptomatic, is that the ACE2 expression levels vary from person to person. So certain people who might not have ACE2 very deep within their respiratory tract or in the alveoli might not experience such strong symptoms, but their upper respiratory tract might still get infected. So if your upper respiratory tract is infected, you could spread by talking, by coughing. The virus only remains in the upper respiratory tract. So one example is that, oftentimes when you have a cold, you would feel that it's just stuck in your throat, and you are only getting a throat infection, and sometimes it will go into your lungs, and you'd have a really hard cough. So it's the difference between those two which cause asymptomatic patients to spread, and sometimes even you don't feel the upper respiratory infection.
Okay, sir. So, the next question is from Hamid Iqbal. He asked, how a diabetic patient prevent himself or herself from coronavirus?
I'm sorry, could you repeat that again?
He asked how a diabetic patient prevent himself or herself from coronavirus.
Okay, yeah. So I
I
mean,
think he asked how a diabetic patient will
how?
prevent.
Okay. So basically, I mean, the regulations would be the same for everybody. That how would you how would you prevent infection? Basically, washing hands, staying away from people, and because in the end, the entry of the virus is all limited to those three or four entry points. Basically, the nose, the mouth, and the eyes. So if you can prevent yourself from doing that. And um, this does not infect that much through blood directly. So with diabetic patients or people who are undergoing dialysis as a result of that, there is little chance of getting infected through the blood. There are more chances of getting infected with other viruses through the blood, which is another issue. But it will basically be the same regulations for everyone.
Okay, sir. So the
But they have
next,
to be extra careful.
okay. So the next question is from Ankita Roy. She asks, is there any chance that the 18-month timeline for development of a vaccine can be shortened, and by how much?
So yeah, so that's a good question. That some of these methods, as I had briefly mentioned, that some of these are well-established vector platforms or vaccine platforms, which already have certain vaccines. So one of the vaccine platforms that I've worked on before is based on a vesicular stomatitis virus, and the Ebola vaccine is actually based on that. So basically, what we do is we take out the glycoprotein of the vesicular stomatitis virus and replace it with a different glycoprotein from a different virus. And the virus that is made or artificially created is now able to infect, now behaves like the virus which has the glycoprotein, or where the behaves like the virus from which the glycoprotein came. So if you replace it with the Ebola glycoprotein, it becomes an Ebola vaccine. If you replace it with the SARS-2 glycoprotein, it becomes a SARS-2 vaccine. So we know that the rest of the virus is the same. So if Ebola vaccine was safe for people, then this will be safe for people as well if we put the SARS-2 glycoprotein in. So this is just one example. So people are trying to build it quickly so that you can just plug and play. So you can just replace what one glycoprotein with the other and then create a vaccine.
Okay, sir. So the next question is from Onushla Chatterjee. She asks, in case of SARS and MERS, antibody titer remained at least for 6 months to 1 year. But in SARS-CoV-2, it remains only for 2 to 3 months. Why is that so?
Yeah, so that's still not known. So it's only like I think last month that it came out that it only stayed for 3 months. But it just happens to be like that because we are just talking about viruses here. But if you take into account a bunch of other things like dust particles or certain type of other foreign antigens, a lot of those are also not lasting more than 3 months or even 1 month. So I mean, there would be some kind of underlying biological explanation, but we don't have that yet. But it just happens to be that this virus is only having this antibody for 3 months.
Okay, thank you, sir. So the next question is from Devdatta Dash. She asked, does BCG vaccination protect against a coronavirus infection?
So yeah, so that's so initially this was found as a correlative study, but I think there's some now there's some studies that are coming out which are looking more into the mechanistic aspect of this. So BCG vaccination, it it is targeted against the bacillus, of course. So it's not going to directly affect, like it's not going to directly affect the virus. But what it does is it creates an atmosphere in the lungs or in the alveoli where this migration cannot happen that rapidly because uh, the immune system has already been triggered in the lungs for the patients who have this BCG vaccine. So the lungs have kind of adapted for that immune response to come in. So it just happens to be acting on the cells of the body rather than on the virus or the bacteria itself. So if the cells are getting more protective, then they will not allow the bacillus bacteria as well as the SARS-2 virus to get in. So it's an indirect effect, but it might work in some people.
So the next question is something like this: that can consuming more warm water prevent this type of virus? And this question is from Hamid Iqbal.
So no, so the amount of the temperature that you would need to kill this virus, that is beyond what the body can tolerate. So it is like you can't really get rid of this virus by warm water. What the warm water does is kind of makes you feel like you makes your throat kind of it soothes your throat. So it's the same thing for if you have a common cold or other viruses. It just feels better if you have warm water. So I mean, if it helps that way, it's fine, but it's not going to directly kill the virus because the virus, as I mentioned, that's all in the alveoli and in the lungs. So the water will not get into the lungs. If it does, that's a big issue.
Okay, sir. So the next question from Sharthak Dashgupta, he asks, is hydrochloroquine really effective against COVID-19 for treatment?
So far we have not seen like most of the studies that found out or tested it, and we have so in at when I left Harvard, like we were still studying that SARS coronavirus. So there was one paper that is still about to come out from some of the work that I did there. So that in that paper we did actually test with hydroxychloroquine because it was like we tested a bunch of different compounds and we also saw no effect. So I mean there were a lot of other studies which showed no effect. So it did not make sense in terms of biological mechanism, like why it would have an effect, because it was targeted towards something completely different. On the other hand, the BCG, of course, as I mentioned, it made sense because it targeted the same cells.
Okay. So the next question is from Taroshri Bhattacharya. She asks, is it possible to develop a proper vaccine against this virus as because the strains are continuously mutating?
So yeah, so that's a good question. So here I'd like to distinguish it from the flu virus. So flu virus, the way that the orthomixer viruses are built is that the entire genome is in little pieces, is in 8 pieces. So when it can actually exchange those pieces between different strains and create completely new viruses. So as a result, that's why the flu vaccine has to be changed every year because it's based on a predictive protocol. So you have to guess basically what strain is going to come out next year and build a flu vaccine on that. So it's not going to mutate as rapidly as the flu or influenza virus, and also coronaviruses have a protein which corrects its mutations to some extent. So it does not rapidly mutate uncontrollably as like a lot of other viruses do. So when we do have a vaccine, it's going to be effective at least for a while. It will take a lot of mutations to actually go away from that virus and from that sequence. And whenever a vaccine is administered, the body produces a variety of different antibodies against a variety of virus proteins. So if one set of antibodies doesn't work, the other set might work.
Okay, sir. So the next question is from Swaraj Ranjan Paul. He asks, so in case of a second time infection, then there will not be any kind of problems to the person as he already has some memory B cells. Is that so?
That is true in general, yes. But then like every individual is different and everybody's immune system is different. It all depends on how much of those B cells or T cells you have and whether those B cells and T cells have actually picked up the original antigen. So if they have not processed the proper original antigen, and if somehow the body has lost those cells, then you might get infected again. So it's very hard to predict from person to person. But later, I mean, as the months come and when there's a vaccine, once you get the vaccine, then people can get monitored and seeing if those B cells, how many people have those B cells and what population has those B cells. Those studies can be done. But right now, like we don't know.
Okay, sir. So the next question is from Rainy Pal. She asks, according to today's newspaper, 72% people are asymptomatic carriers of COVID-19 in our state. Can COVID-19 be fatal in case of asymptomatic patients?
I'm sorry, the last part I missed it. Can COVID-19 be
Fatal in case of asymptomatic patients?
subtle? Yeah.
Okay.
Yeah, yeah. I mean, it could definitely be subtle. So you might not even you might only have like one of the symptoms that are typical. So you might just have a little bit of scratchy throat for 1 or 2 days, and that would be it. So if I correct answered it correctly, if I understood the question.
Okay, sir. So the next question is from Tonmal Rashgupta. He asks, do citrus fruits provide a proper detoxification against COVID?
I'm sorry. Could you
Citrus
repeat that,
fruits,
please?
citrus fruits.
Citrus fruits. Oh,
Yes.
no. Yeah, it doesn't have a correlation because if it like everything that goes in through the stomach, like it won't affect the virus because it's mostly colonizing in the respiratory tract. So if you're eating anything or trying to get immunity through that, it will generally increase immunity. Like vitamin C is it's an important component of the immune system, but it's a general effect, and it will not necessarily protect you against this virus particularly.
Okay. So the next question is from Tridip Shaha. He asks, does COVID-19 present any unique risk to pregnant individuals?
So pregnant individuals, yeah. So that's a good question because I think it was yesterday that I was reading this that there have been some reports which have shown that babies just after birth they have gotten infected and they carry the virus and they tested positive. But it could most of those cases so far had been thought that yeah, it could happen inside a room, so the mother and the child and everybody is in the room, so it could be a direct transmission right after birth. But there was one paper which just recently published which shows that it could be a direct transfer from mother to child. So, but it's still one report, and it's still not clear whether that's the real phenomenon. So once like more reports like this come out, we'll get to know.
Okay, thank you, sir. So the next question is from Shubham Nondi. He asks, how can we design a vaccine ASAP for such a rapidly mutating virus? And can newborn and the aged people who have higher susceptibility towards the virus be specially addressed?
I'm sorry, I missed the last part. Yeah, I think it's very faint, the sound that I'm getting from your
Oh,
microphone.
am I audible now? Can you hear me now?
Yeah, but it's really faint.
Oh, can you hear me now?
Yeah, I mean I can. It's faint, but yeah, please go ahead.
Okay, so I'm repeating the question. The question is, how can we design a vaccine ASAP for such a rapidly mutating virus? And can newborn and the aged people who have higher susceptibility towards the virus be specially addressed?
Yeah, so I think it was like related to the question that we got before. So it's like similar to the flu vaccine that it's not going to be that hard to design a vaccine because it does not this particular virus does not mutate as rapidly as the flu virus or some of the other RNA viruses.
Okay, sir. So the next question is from Swaraj Ranjan Paul. He asks, suppose a vaccine is developed in any of the European countries, will it be equally effective for the people of our country?
Again, I'm sorry, I just can't hear.
Oh,
Um,
I'm so sorry, sir. I think there is some net connection problem. So, can you hear me now?
yeah, this is better. Yeah,
Okay, so the question is like this. Suppose a vaccine is developed in any of the European countries, will
yeah.
it be equally effective for the people of our country?
It should be effective because the vaccine is actually directed towards the virus, not towards individuals. So if the virus is the same, then it will be the same for every person.
Okay, so the next question is from Gargi Bhattacharya. She asks, how will high BP patient get prevented from coronavirus?
So yeah, that's an important factor. So I didn't go that much into detail about the therapeutics that are also being developed. So some of those are actually dealing directly with the symptoms. So high BP patients are at a risk for sure for this virus. So oftentimes, as I showed in my presentation, the angiotensin it causes vasoconstriction and increase of blood pressure. So often, for a lot of COVID-19 patients, the treatment that has been investigated is ones that cause vasodilation, or it prevents vasoconstriction. So trying to prevent that effect of uh, the arteries or the veins contracting and causing high blood pressure. So that might relieve some of the patients.
Okay, sir. There are many other questions. Are you willing to take it?
Yeah, I'm totally fine.
Okay, thank you, sir. So the next question is from one of our students, Rajveer Day.
Mhm.
He asks, sir, why weren't we using AC2 blockers to stop the virus, and can we make temporary AC2 blockers and which have temporary effect on body? In that time, we can stop the virus in meantime.
Yeah. So that is one of the therapeutics again, like which I didn't go into detail. So a lot of ACE2 blockers have been investigated already. Some of them have been effective, and some of them had been used to control blood pressure before. So those are some of the drugs that are coming into the market where they can repurpose it and use it for the virus. But the problem is that once it gets in, then the function of ACE2 is over. So after that, whatever it does, there has to be something else that can stop the virus.
Okay, there is another question related to this. It is asked by Aratrika Ghoshal. She asks, is it possible that the virus might bind to the AC2 receptor of some other organ of the body after its entry into the body?
Yeah. So whichever organ it infects, it will have to bind to the ACE2 receptor. And in fact, there's another. So I skipped that part, but it would be interesting to mention it here. There's an enzyme uh, in a lot of cells of the body called TMPRSS2. This enzyme is necessary for the virus to get activated because it kind of cleaves the glycoprotein and activates the virus, and the virus can now get into cells. So basically, it's whatever cells have ACE2 and this TMPRSS2, these two together, the protease and the receptor, these are the cells that are most susceptible. So yeah, I kind of left that part out.
Okay, thank you, sir. So the next question is from Debayani Chakraborty. She asks, how do you think the reservoirs for these zoonotic viruses change with time? If this virus really came from bats, what change do you think might have occurred in the structure to help them infect us?
So what the change that has happened is in there are about I think 14 residues or 14 amino acids in right at the top of the spike protein, which uh, which bind the receptor. So there are out of those 14 residues, I think 4 or 5 residues have changed from the bats, and as a result of those 4 or 5 amino acid changes, it can now bind the human ACE2 receptor. So bats also have ACE2. Armadillos is another intermediate animal that's been studied. So they also have ACE2. And based on how those ACE2 sequences are, it's been suggested that those changes are what caused the binding difference.
Okay, sir. So the next question from Ritika Banerjee, she asks the question is that is it possible to make a vaccine which is effective against all type of virus?
All types of coronavirus, I'm guessing.
Yeah.
Yeah, yeah. So I mean, it is possible. But it will require a lot of design. So already, so coronaviruses are something new, as you would have seen that like things only started getting uh, studied after 2003. But flu has been studied for a long time. So there's a big push to try and get a universal flu vaccine. So something that will work against all the flu strains, and we won't have to make a new vaccine every year. So it is possible, but it is really difficult.
Okay, sir. So the next question from Orphan Day, he asks to prevent further pandemics. What is your take on expansion of constant monitoring or surveillance and imposing strict regulation for various labs conducting research to prevent accidental exposures?
Yeah, so as I mentioned in the last part of it, the viral surveillance, uh, whenever certain strains that are found that are very close to so when people are looking right at the sequence level. So when I just mentioned that those 14 amino acid residues, for example, if you find a virus which has 13 residues which are similar and one residue which is different from the human virus, so you would think, oh, this is a very high chance of mutation. So those viruses, what scientists should do is to clone those viruses or isolate those viruses, take out that sequence, and preemptively create a vaccine for that. If once the platform is created, for instance, like right now, there's no coronavirus vaccine, but once it's created, it will create a template based on which you can plug and play all of different kinds of coronavirus vaccines. So the next coronavirus that comes up could then be designed based on that uh, base vaccine.
So one last question is from Priya Shaha. She asked, previously we were told that mainly aged people are more susceptible to infection. But with passing days, people of young age are also dying. Why is it so?
So one of the reasons is probably because this virus, as I mentioned in my presentation, this current virus that's circulating is a mutant, and that has changed with a single amino acid residue, which has made it 10 times more infectious. So now a lot more people are actually getting infected due to this virus. So that could be one of the reasons. Or the other reason could be just that now we have way more testing and a lot more people are getting tested. Previously, people are at risk and older people were, or people who had actual symptoms were getting tested because there were not that many tests.
Okay, sir. Thank you so much for clearing all the doubts of our participants. We are really glad to have you among us for such an informative presentation. So now it's over to you, Anushriti.
Okay. Thank you, sir, for this extensively informative interactive session. There was enormous questions, and thank you again for answering them all in detail with patience. You really cleared all our confusion and perspective towards COVID-19 pandemic. Now, I would like to request Dr. Shashwati Gayan, Assistant Professor of Department of Microbiology and of Vijayagarh Jyotish Roy College, to proceed for the vote of thanks. Ma'am.
Good morning, everyone, and good evening to Dr. Shayantan Bose. We have reached almost to the end of today's session and also to the end of the lecture series on In Search of Origin, Prevention, and Possible Cure for COVID-19 organized by Department of Microbiology in collaboration with Internal Quality Assurance Cell, Vijayagarh Jyotish Roy College. At the very onset, I would like to thank Dr. Shantan Bose for delivering such a wonderful, interesting, and informative lecture on COVID 19 and our future. Thank you, Dr. Bose. Thanks to our respected principal, Madam Dr. Rajyashree Niyogi, for her constant support and motivation. Thank you, Madam. I'm also thankful to all the faculty members of the Department of Microbiology, especially Dr. Proshenjit Das and Dr. Shilajit Borua, and to the MSc students of our department and research scholars who have helped us in organizing this webinar successfully. Finally, I would like to convey my sincere gratitude to all the participants for their valuable presence and support, which is highly appreciable. Thank you all, and hoping the same response from you in our future webinars as well. Stay home, stay safe, and take care. Thank you all.
Thank you, ma'am, for your vote of thanks. Now we have come to the end of today's session. Thank you all for your participation in this international webinar lecture series. Don't forget to drop down your valuable feedback in the link provided for procurement of e-certificate that would be delivered to your mail. Thank you all, and thank you, sir, again for your valuable time.
Thank you very much again. I mean, I would, as I said, it was a wonderful opportunity for me, and the questions were really good. So I'm so happy that I would be able to answer that. And and it's always a pleasure to talk to students.
Thank you, sir. We're really grateful to have you.
So, Dr. Bush, it's now over. Very informative and wonderful talk you have presented, and hope that we'll remain in touch for
of course
scientific endeavors. And so it's I suppose it's a good night for you. So
Yeah.
so that's fine. Thank you.
Okay. Thank you. Please stay safe and have a
You too.
good day. Yeah.
Yeah.
Bye.
Yeah.